Okay, so we continue. So the degradation, now we enter into the degradation of lipid. Seafood roughly can be divided into 4 categories. The first one is lean species, means that it doesn't have a lot of lipid, which constitutes below 2% of the total body fat. Low fat species, up to 4%. Medium fat species, up to 8%. And fatty, which is more than 8% of the total fat. However, different parts of the fish or the seafood can also have different amount of fat. For example, the amount of fat at skin, viscera, which is below the stomach, or dark or white muscle, they all can be different. So this classification is not very accurate. It's just one of the guidelines that is used to show the type of the species. Aquatic animals are rich in long-chain polyunsaturated fatty acids, especially at cold areas, probably because of their diet, okay, or they need to protect themselves from the temperature and so on, I don't know. However, because of the presence of a lot of long-chain PUFAs, this presents the oxidation problem, means that the lipid can easily oxidize. Unsaturated and structure of lipid that has bigger surface area, they are easily exposed to pro-oxidants. Pro-oxidant is something that is opposite of antioxidants, means that they can oxidize the compound that they come in contact with. Okay, example of the pro-oxidants can be heme proteins, which contains in the red blood cell, radicals. Okay, if human beings, we can get this radical from the formation of energy or formation of ATP, or the environmental factors, for example from the sunlight, okay, which because when the UV hits the biological molecule, they can produce radical. The presence of iron, copper in the body, which usually present in the metabolically active dark muscle. They can be accelerated at low pH, and also when the meat is minced together. So when the meat is minced, the pro-oxidant and the oxygen from the environment, they can mix together and accelerate the lipid degradation. Or during the thawing, because the thawing apparently increase in iron and copper and lipase activity in the seafood. The lipid degradation can lead to rancidity, okay, another word for unpleasant odor and texture. I'm not really sure how the taste is because I never eat the rancid seafoods, but apparently it is very strong. So we know that the seafood has gone bad. It can also change the texture of the seafood. For example, it can cause muscle toughening, lost water, holding capacity, protein denaturation, and also the color change. How to avoid from the rancidity of lipid? The first thing we can do is we can drain the blood of the seafood. So we reduce the heme or pro-oxidants that can come in contact with the lipid. We can reduce the tissue disruption during storage, because if the tissue is disrupted in the seafood, it can release all the pro-oxidant and lead to the lipid degradation. We can wash the fish fillet or the minced fish to remove all the pro-oxidant. We can remove the dark muscle. So when we cut the fish, we can remove certain place where we can see a lot of blood is accumulating. We can try avoid the oxygens, means that we can try package the fish or the seafood in a vacuum, or add carbon monoxide to reduce the heme activity. We can also add certain anti-oxidative components that can nullify or neutralize the pro-oxidants. Pigments is a structure that gives the color to the seafood. So logically, we want to retain the color of the seafood so it looks fresh. Okay? The main one is hemoglobin and also myoglobin in form of heme. Both of them contains heme. Heme is red in color. but usually the one that is retained in the muscle protein is myoglobin. Hemoglobin, actually the one that is in the blood. So when you drain the blood, so the color is gone. What retains is myoglobin. This one usually in the warm-blooded seafood, but in cold-blooded shellfish, different kind of blood is present with fish, hemocyanin. Sometimes the color is blue. Okay? It doesn't really have hemoglobin, so it's really special. And also in certain fish, for example, salmon, they can contain carotenoids that give certain colors. For example, pink color. Okay, degradation of the pigments can lead to epithelial discoloration. Okay, for example, fresh squid shows dark red brown pigment or monochromes because rigor mortis causes contraction and give pale and light discoloration. Fresh squid usually they show this dark brown pigment, but if they die, they can turn to pale or light coloration. This pigment can be retained by freezing or dehydration, but if we do any improper storage, it can cause red discoloration. However, certain freezing may cause subcutaneous fish yellowing flesh. Okay? The fish color can turn into yellow. So probably there is a strategy to reduce this yellowing of the fish. For hemoglobin, like I said, it is less important than myoglobin for the coloring because usually most of them are drained when we remove the blood. However, it may give green hues when combined with hydrogen sulfide during meat decomposition. I think it's here. Pigments. Okay. You can see it can turn into greenish color in the meat. Hemocyanin, usually they are copper rich instead of iron like hemoglobin. However, it is not really well understood. But the blood of these creatures, hemocyanin, they are very valuable in pharmaceutical area, okay, because they can be used as a carrier and so on. I think I can show you hemoglobin. Hemocyanin. Okay, you can see the color can be blue or green, I don't know, depending on the creatures or animal. Okay, myoglobin, discoloration of tuna during freezing due to the formation of metamyoglobin formation. Greening of tuna can also occur due to the cooking methods because of the presence of oxidizing agent. Carotenoids in seafood may be easily oxidized by lipogenase-like enzyme. Black spot in shrimp during postmortem storage can be caused by phenol oxidase. Okay. Another term for that is melanosis. Melanin. So this is the example of different color in meat because of the exposure to the oxygen. So exposure to high oxygen can cause this kind of color. Low oxygen can cause, I don't know, this is green. Okay, and no oxygen can give red brown color. Okay, if you want to measure the freshness of the seafood, you can use these biochemical indices. Okay, these are all the metabolic changes, not microbial indices. This is the metabolic one. For example, the lactic acid formation, so lower pH, the nucleotide catabolism, okay, means the formation of degradation product of ADP. ADP comes from ATP. Degradation of myofibrillary proteins. Okay, we can measure this one. Collagen degradation, which lead to the honeycombing formation and mushiness. Dimethylamine formation, TMAO to DMA during freezing. Okay, we already discussed this one before. Free fatty acid accumulation due to lipid degradation. Okay, because when the lipase is active after the death of the seafood, it can break the lipid to free fatty acid. And finally, the tyrosine accumulation. Okay, because the drop in pH can lead to activation of proteases that can break the cell and release the tyrosine from inside the cell. Okay. Changes during freezing. The first one is the formation of ice crystals that can disrupt the pH, osmotic pressure, and ionic strength. Second, changes in the physical state of water and the presence of lipid induces protein denaturation, which can lead to textural change. Okay, of course, because the texture is depending on the shape of the protein. Okay, it can be fibrous, tough, spongy, less juicy, and rubbery. It can be caused by one or more of the following factors. Okay, the first one, again, the formation of the ice crystal. Therefore, it's recommended to use a faster freezing method compared to slower or longer freeze, okay, to reduce the formation of the ice crystal. Second is the dehydration effect. Freezing removes water from the protein and disrupts the hydrogen bonding. The third one is increase in solute concentration. Salt will increase in the unfrozen matrix, lead to the pH change. The fourth one is interaction of protein with intact lipid and free fatty acid. Formation of lipoprotein complexes that may change the muscle texture. This is all happening during the freezing. Interaction of protein with oxidized lipid. Proteins becomes harder and insoluble. Okay. Next, freezing can also cause lipid degradation. And finally, the DMAO, enzymatic degradation. Okay, so usually the seafood, because they are not very, the shelf life is very short, we try to process them into something else. Okay, for example, the process involved can be salting, means that we add salt. Second is fermenting. Okay, we can marinate them, drying, or smoking, so that we can increase the shelf life and develop sensorial properties. Okay, the main way of developing the sensorial properties and increasing the shelf life is by reducing the water activity. Okay? For example, when we drying or when we salting, we remove the moisture from the seafood. We also can reduce the microbes by heat, preservative, and antibacterial compounds. Okay? For example, you can add nitrates, so the kind of preservative inside the seafood to make sure that the shelf life is longer. However, the heavily salted fish may lose more water due to osmosis. The texture can be tougher. It can develop less flavor, mushy texture if calcium or magnesium are present. Okay, probably certain kind of salt contains this ion, and over-ripening can lead to excessively soft product of the salted fish. Capsaicin family and trypsin-like enzymes can contribute to the sensorial properties of fermented and dried seafood. Okay. So it has been shown that if we add or undergo these processes, this kind of protein is the one that can give the unique taste or sensorial properties of the seafood. Okay. For the thermal processed seafood, elevated temperatures may activate enzymatic activity that lead to the softening of the fish texture. Okay, so when we cook them, they can be softer or tender. Thermal treatment or pre-cook can coagulate protein, remove impurities, and inactivate enzyme for easier handling. Okay, so for example, the canned sardine, they cook first so that they can undergo all these processes of removing the coagulated protein, impurities, and so on. Crab meats are pre-cooked to remove the flesh from the shell, otherwise it is very hard to separate from one another. Mild heating can be used in dried seafood to inactivate certain enzymes that can lead to the lower quality of the seafood. Alright, so that's all for the fish. We will take a look at the presentation of your colleagues later on for a more detailed presentation of fish and seafood. Okay, thank you.